getting caught the same as Gharial and Tomastoma, because they got that long snout that goes through the mesh, and as soon as they pull back, they catch the mesh on their teeth, and then the next thing they roll it up. But certainly, when we were trying to catch them, we had to vary a whole bunch of things to work out the best way of catching them. And if you undo that, you might be able to work out the worst way of catching them. It's just, I don't know whether anyone's looked at the nets. That was really my question.
Can you hear us, Anna?
Okay, thank you very much. Can you hear me?
Yeah, we can hear you.
Can you hear me?
Yes, we can hear you well. Go ahead.
Okay. Yes, I can, yes. Okay, so thank you for the question. Oh, I think I've got two things on at once here. Can you still hear me, yeah?
Yes.
Yes, okay, so a lot of the data that I wasn't able to present was the work we did. We had a whole range of questions which were actually asking about fishing techniques. And part of the problem is, and it's a vicious cycle actually, is that because fish stocks are going down, people are reducing mesh sizes beyond smaller than the legal limit and increasing the length of their nets. And they're catching smaller and smaller fish, which is obviously going to have a very negative impact for the sustainability of fish populations, as well as making things worse for crocodiles. So we're very well aware of that problem, and we're working with the Brigade de Pêche, the fishing brigade, as well, and we've shared these results. Actually, the response we had from fishermen was very positive. They were actually very glad that somebody was interested in the problems they were facing, which were to do with fish stocks, but also obviously included losing nets to crocodiles. So the smaller crocodiles get caught in the nets and are then usually consumed or sold. The larger crocodile, like the Nile crocodile, doesn't tend to get caught because it destroys the net and goes away with it. But either way, it's bad for crocodiles and fish. So we do have some information on fishing techniques, and nets are not the only one. Another technique which is illegal but is quite widely used is something called tape-tape, where they use wooden paddles to beat the water to help drive fish into nets. And they also reported that they thought that this was had a negative impact on crocodiles as well.
So just to add a brief thing to that, there was another Gabonese researcher, and I don't know if you know Regis Kemakema, but he did his work on this issue, like if they could realign the net, like something about the fishing techniques in an area just to the south of Anna's site. And they basically found, it's been a while since I looked at the study, but if I remember correctly, they found that even just like moving the net 2 meters away from the edge of the vegetation significantly reduced mortalities without a significant change in catch from the fishing catch. It's just a question of really getting fishermen to take that up. But this might be something, Anna, if you reach out to Regis, he probably has his study and you guys can talk about it to figure out another thing to work on potentially, some hypotheses to test. And then also just Graham,
yes.
that's Megalop's leptorhynchus in Gabon, not cataphractus.
Yes, exactly, and we're aware of his work, indeed, showing that putting the nets, as I mentioned, I think perpendicular to the bank doesn't significantly impact fish capture, which is a good thing for fishermen, but does help crocodiles and other aquatic creatures quite a lot. So it's a case of trying to get that implemented and working with local people so they believe that changing technique is actually going to work both for them and for the wildlife. Thank you.
Okay. All right. Thank you so much. Can you guys just give the speakers all just another round of applause just to thank them? It's been a couple of really good presentations. Thank you so much for that.
Okay, welcome back, everybody. We are now going to start the session on crocodilian biology, and our first speaker is going to be Francois Clarac.
Thank you. Thank you. I'll just turn out the sound. And now, slideshow from the start. Yes, perfect. Okay, good. Sorry. Okay. Oh, wait. That's right here. That's just not. There we go. Right. That's better. The slides are not. Well, we'll do it again. Yes. Hello, everyone hears me? واحد زوج. Yes, it works. Hi everyone, I'm François Clarac. I'm both lecturer and researcher in Sorbonne Université. I teach and investigate both in the fields of... Okay. Time out?
No, keep going. You're fine.
Okay. Okay, both in zoology and paleontology. So I'll be here very happy to share with you my results, which I got from a collaborative framework with Zoological Park of Paris, with French Guiana Zoo, and of course, Crocodile Park. So this work is about finding out the adaptive thermoregulation strategies in the evolution of crocodilians, with a special focus on the Amazonian caimanes. What happened? It used to work. Okay. If it works here, no. Exactly. Matt, what have you done? Okay. Now it does work. It's fixed. Thanks. Okay, so I just say we're making here a focus on the Amazonian caiman species. So there are basically 4 species: the black caiman, which is by far the biggest one; the 2 species of dwarf caimans, which are the smallest species in the world; and the spectacled caiman, which has an average size and is not only located in the Amazons, since we find him in the northern territories up to Central America. So, spectacled caimans live in all types of water bodies, medium, large, and not exclusively in the Amazons, of course. Meanwhile, the dwarf caimans live in small water bodies that are usually covered with by the canopy. And the black caimans are found in large water bodies, which are usually oxbow lakes. So these oxbow lakes are shaped up by both the erosion and the sedimentary deposits along the river channel that ends up isolating an oxbow lake. Then the water will go down in the oxbow lake. The plants will just end up covering the place, which will be just very sunny with nice jungle surroundings, and will cause black caimans, just like this one. Okay, so that's about the landscape. Now, what is thermoregulation in crocodilians? Basically, how does it work? Okay, the point of thermoregulation is to maintain the vital organs, so mostly the brain and neurosensory tissues, as closer to the optimal temperature. To do so, crocs have very well vascularized skin, but this vascularization is pretty much uneven. It's almost only located in the pits of the ornamented osteoderms and in the skull. So if you take a look at an albino specimen, it's very easy to see. You will see that over the dorsal shield, you will see all these little red spots. Basically, they're blood vessels. Meanwhile, the rest of the body is white because there are just no blood vessels. Interestingly, when you take a look with an infrared camera at the same specimen, you will see a match between the temperature distribution and the blood vessel distribution. Then, thermoregulation is controlled by both the heartbeat rate and the vital dilation. And a very interesting point is also the shifting routes between veins and arteries. As you could see in some presentations before, the cardiovascular system in crocodilians is very complex. So here, for instance, we will have in blue pathway that drives the blood through the brain case, while the red one will avoid the brain case. So what do we do if you want to learn about heat transfer then? So what we do first is that we, of course, as I showed before, shoot a specimen with an infrared camera, so you just can see the temperature on the surface of the body. And then you just have to measure the internal temperature. Using, for instance, here, we used a temperature sensor that we hide in the meat. So the animal will just swallow it, and we will be able to track remotely the body temperature with a monitor that here, Margot of the Guyane is just showing. So she's showing of what we know the first temperature record in the black caiman, which we obtained by hiding the monitor in the surrounding of the animals. That's why here I just built a little zip line in order to record the body temperature, which is recorded by the sensor inside the stomach of the animal. But what are the limits of this method? Well, regarding infrared camera, we know that they just don't work through water. That's one thing. It's also the case regarding thermal sensor. But one other main limitation is that the animal must accept feeding. So I think you should forget about that in the wild, of course. Other issue is that the range of the monitor antenna will decrease with body mass, which again can be a real issue when you work with crocodilians. But basically, this is the kind of result you will get. For instance, here you will see in black the air temperature of the day that is also recorded by a sensor, and you will see that the body temperature of the spectacled caiman will follow the air temperature evolution, so you will see the peak by the end of the day. And meanwhile, when you observe the specimen with an infrared camera, you will see this red spot over the dorsal shield, which shows that the temperature, the external temperature has been gathered this way. And then the body temperature will go down through the night until it gets a colder point here in the early morning and starts again a new cycle. So that was about the spectacled caiman, but what about the black caiman? Well, as I told you before, Margot recorded 2 temperatures, and that's pretty much so far what we got, for a very simple reason, is that we set the monitor as close as possible to the body, because the black caiman is has a large body, so we have to get the monitor really close with the zip line, and so far I was very satisfied with that, but she decided otherwise. Yeah, you know crocodilians. So at some point, she turned around and she bit the monitor. So then I ended up with 3 issues. First issue, it was the end of data acquisition. Second issue, I was out of 2,000 euros. And last issue, last but not least, we had to make her let go. And she bites very strong. So at the end, I got back my monitor in this, like on this picture, kind of. But, you know, shit happens, and, you know, I still had an infrared camera, so there was some still work to do. With black caimans, especially because one specimen was just near this one, was living all the time. 2 minutes left? Okay, I make it quick. So was just staying in inside water, so I could follow, I could track this black caiman with the infrared camera. So what could I see? It was that the skin temperature will overtake the air temperature during the warm day, the warm hours of the day. So that was a very interesting result, to see that besides it was in water, it was still thermoregulating. Also, some results, I'm just making this quicker. The results we had further on in the dwarf caiman was very different, because the body temperature of the dwarf caiman was just maintaining 26 degrees all day long, whether it was in captivity or in the wild. As you can see here, they will just hide in the plants or stay in water below the trees during the day, the warm hours of the day, so the body temperature won't increase, neither the skin temperature. Okay, yeah, you can see here the pond is in blue, yellow, low temperature, and that's where they stay. Okay, so here you have this main contrast between the black caiman and the dwarf caiman, although they live at the same place. Okay, so this is, by the way, very consistent with previous results that were found by Zilka Campos in Brazil, who had some study, who made some study independent on the dwarf caimans, who always show the same temperature, whether it's summer or winter. Okay, so we will go to the conclusion. In conclusion, we can see that black caimans and spectacle caimans are adapted to forest habitats with spotlights, whereas dwarf caimans are adapted to forest habitats without spotlights and stay in their canopy. So I will switch on that because we don't have time. And the real conclusion is, we think that the standard thermoregulation in crocodilians is active thermoregulation, which we find in almost all crocodilians, but in dwarf caimans, we'll see thermoconformism, which is probably an adaptation to live under the canopy in equatorial forests. And the wonder is it the same in dwarf crocodilians, as you can see here in Osteolaemus, and it will explain why they could move from forest dwelling to cave dwelling in the Central Africa equatorial forest. And I will switch that, and I will just go to acknowledgements. So I just want to thank my collaborators, and especially Luc and Ariane Marinetti, who organized both this Congress and helped me get this result by hosting my studies in Croco Park. Thank you for your attention.
Okay, a brief announcement before our second speaker. Someone left some meds in the restaurant. They're up here, if you would like it. They're over here. And our second speaker is Christine Coumont, and she's going to talk to us about the social dynamics, and in English, right? In French, and so we're going to have some translation. Thank you.
Thank you. Good afternoon, everyone. My name is Christine Coumont. I'm from Côte d'Ivoire, and we're gonna go through my study on social dynamic of crocodile of the genus Mekistops in Tai National Park and Luango National Park. But the talk will be given in French. Jusqu'en 2014, le genre Mekistops était considéré comme monospécifique. Et des études récentes ont montré qu'il existe deux espèces distinctes, dont l'une, Mekistops cataphractus, localisée dans la zone forestière d'Afrique de l'Ouest, et Mekistops leptorhynchus dans la zone de l'Afrique centrale. La population de Mekistops leptorhynchus est plus ou moins viable, mais le statut de l'espèce est en cours de révision, avec de fortes probabilités qu'elle soit en danger ou du moins vulnérable. Quant à Mekistops cataphractus, les populations sont menacées et le statut de l'espèce est en danger critique avec moins de 500 individus en milieu naturel.
So the slides say everything. 500, probably less than 500 individuals of Cataphractus left.
Les menaces auxquelles font face ces espèces sont nombreuses, mais les plus importantes sont la destruction de l'habitat avec la déforestation et l'orpaillage clandestin et la mortalité par prise accidentelle dans les filets de pêche. Il y a également le changement climatique qui impacte gravement la survie de l'espèce. Pour assurer la survie de la pérennité de telles espèces, la Commission de suivi des espèces a préconisé plusieurs mesures, parmi lesquelles l'augmentation de la population en milieu naturel par réintroduction d'individus. Mais pour que ces mesures puissent être un succès, il faut avoir de bonnes connaissances de la biologie et de l'écologie des espèces concernées. Ce qui n'est pas le cas du genre Mekistops, car les espèces concernées sont parmi les moins connues au monde. C'est dans l'optique d'assurer une conservation à long terme des crocodiles du genre Mekistops que le projet Mekistops a été initié en Côte d'Ivoire. Et il a pour objectif d'améliorer les connaissances sur les espèces Mekistops cataphractus et Mekistops leptorhynchus. Ensuite, promouvoir le potentiel de l'élevage en captivité de Mekistops cataphractus aux eaux nationales d'Abidjan. Et enfin, augmenter la population en milieu naturel de ces deux espèces. Nos travaux ont été réalisés au Parc national de Taï en Côte d'Ivoire pour Mekistops leptorhynchus et au Parc national de Luango concernant Mekistops cataphractus au Parc de Taï et Mekistops leptorhynchus au Parc de Luango. Nous avons utilisé la télémétrie pour suivre les individus afin de collecter les données. Nous avons bagué des mâles et des femelles des 3 classes de taille, c'est-à-dire juvéniles, subadulte et adulte, et les tailles ont varié entre 1 et 2,81 mètres. Pour le radiopistage des individus, nous avons utilisé la méthode de confirmation visuelle qui est le homing ou la méthode de triangulation lorsque l'accès à l'animal était difficile. Après collecte de données, nous nous sommes posé plusieurs questions afin de trouver des réponses pour nos objectifs. Donc la première question, c'était de savoir quelle est la proportion que chaque individu partage avec ses congénères. Et 3 cas de figure pouvaient se présenter. Le cas où l'individu partage une faible partie de son habitat, soit une moyenne partie ou une très grande partie de son habitat. Les résultats indiquent que, aussi bien pour Cataphractus que Leptorhynchus, plus de 80% des individus ont partagé une bonne portion de leur habitat. Nous en concluons qu'il existe une absence de territorialité stricte au niveau de ces deux espèces et donc il est possible de réintroduire plusieurs individus dans un habitat donné. En regardant de plus près la classe démographique des individus en interaction, nous avons remarqué que les classes de taille, les individus de classes de taille différentes, c'est-à-dire ici juvénile, subadulte, juvénile adulte et subadulte adulte, étaient les plus fréquentes. Donc il est possible dans ce cas-là de réintroduire des individus de différentes classes de taille dans un même habitat. Au niveau du sexe des individus, nous remarquons qu'il y a une plus grande proximité spatiale entre les mâles et les femelles, ce qui nous amène à dire qu'il est possible de réintroduire dans le même habitat des individus de sexes différents. Ensuite, nous nous sommes posé la question de savoir le comportement des individus qui interagissent au sein de la zone commune. Donc, 3 cas de figure également se présentent. Soit l'individu occupe très peu la zone commune, soit il utilise de façon aléatoire ou il utilise très fréquemment la zone commune. Et nous avons classifié les différentes paires en interaction selon la grille suivante. Le premier cas, c'est le cas où les deux individus utilisent très peu la zone commune jusqu'à le cas, jusqu'au cas où les deux individus sont très fréquents dans la zone commune.
Okay, I think this is clear from the slides. Should I translate? Basically looking at the pair association within the shared area, and that shows the continuum possible from avoiding to share it to enjoying sharing it and all the in-betweens.
Et les résultats ont montré que les cas de figure où au moins l'un des individus préfère la zone de chevauchement à celui où les deux individus préfèrent la zone de chevauchement sont les plus importantes. Après cela, nous nous sommes posé la question de savoir quand est-ce que les individus utilisent la zone de chevauchement. C'est vrai que les deux individus peuvent préférer la zone, mais est-ce que l'utilisation est faite de façon simultanée ou alternée? Ici, c'est le cas où un seul individu est présent pendant que l'autre est absent. Et l'autre cas, c'est là où les deux individus utilisent de façon simultanée la zone de chevauchement. Et selon nos résultats, les individus utilisent de façon alternée la zone de chevauchement, ce qui nous amène à dire que les crocodiles des deux espèces ont développé une stratégie de partage des ressources disponibles dans la zone de chevauchement tout en évitant les rencontres conflictuelles. Donc, au vu de tous ces résultats, nous pouvons déduire qu'il ne faudrait pas réintroduire dans le milieu des individus de classe de taille homogène ou de sexe homogène, mais plutôt privilégier la réintroduction d'individus de groupes hétérogènes constitués de différents sexes et de différentes classes de taille.
Right, so in light of these results, their recommendations are that it's best to not reintroduce animals of same size and same sex, and it's better to reintroduce animals of different sizes and different sex because they tend to use the area alternatively instead of simultaneously.
You didn't translate the results.
Okay. Au-delà de ces résultats, nous envisageons continuer à améliorer nos connaissances des espèces concernées en étudiant le comportement reproducteur des femelles pour savoir leur mouvement au cours des différentes saisons et aussi la sélection spécifique de l'habitat. Nous voulons également conduire une étude de télémétrie en dehors des aires protégées, puisque celle-ci a eu lieu dans 2 aires protégées, pour comprendre de façon globale le comportement des individus. Et enfin, pour mettre à disposition des données scientifiques pour la mise en place de projets de réintroduction, nous voudrons bien modéliser les habitats disponibles pour les espèces concernées pour les programmes de réintroduction futurs. Au terme de cette présentation, je tiens à remercier toutes les structures qui ont bien voulu nous accompagner dans la réalisation de ce travail et de façon spéciale le groupe de spécialistes des crocodiles pour avoir permis notre présence ici aujourd'hui. Je vous remercie pour votre attention et je suis disponible pour toutes les questions. Merci.
For our judges, we just had a student presentation, and I'm going to ask you to remind me when you come up here to let me know that you're a student. Our next speaker, who's eager, because she's up on the stage well before I called her, is Brenna Dakin from Crocodile in Florida, and she's going to tell us about nesting in Acutus.
Hi, as Venetia just said, my name's Brenna Dakin. I'm a researcher with the University of Florida Crocodile, and today I'm going to be talking to you about a project that we've been working on, examining the influence of substrate type on the nest microclimates of American crocodiles in Florida. So just to give you a little bit of background, American crocodiles were listed as federally endangered in the United States back in 1975 due to overexploitation and habitat loss for nesting. We began monitoring nest efforts in Florida in 1970 and have continued these efforts annually since then, as nesting effort and success are important parameters when monitoring the health of a population. The results of this long-term research have shaped management decisions, which led to the downlisting of the species to federally threatened in 2007. As the nest microclimate conditions play an important role in nesting success, we were interested in looking at how these conditions varied in nests laid in natural habitats versus human-altered habitats. So here you can see the number of nests that we've had over the last 55 years. These dark blue bars here represent nests found in Cape Sable. This area has served as the most important area for American crocodile nesting in Florida over the last 18 years, making up 41 to 66% of our annual nests over that time frame. So because this area is so important for nesting in Florida, we were interested in looking at nest microclimate variation within this area. So here you can see a map of Cape Sable. All of the areas labeled are the locations where our crocs nest every year. So there are two nesting substrates that they'll use in this area, sand and marl. So Clubhouse Beach, East Cape Creek, East Cape, Middle Cape, and Northwest Cape are all beach habitats which have the sand substrate, while East Cape Canal and Homestead Canal are berm habitats with the marl substrate. These two canals were dredged out in the 1920s in attempts to drain the interior of Cape Sable to use it for agriculture. Since then, they have plugged both of these canals to prevent further bank erosion and help prevent saltwater intrusion into these areas. So we chose to examine East Cape Creek as our more natural sandy habitat and East Cape Canal as our human-altered habitat in this area. So here you can see the exact locations of the nests which we instrumented with data loggers. We put data loggers in 2 sand nests and 3 marl nests in 2025. So here you can see some pictures showing the different habitats of the beach habitats and the canal marl habitats, and some examples of some nests within these. The sand out here consists mostly of fragmented seashells, while the marl is composed mainly of clay. The marl is much more hard and compact than the sand, and just to kind of showcase this a little bit more, I have a little video to show you. Also just for fun, because it's cute. One of the problems we do see with this substrate as well, because it gets so hard, is that if mom does not dig them out, the babies aren't able to get themselves out, which they can do with sand. Of course, they don't always do that because they're lazy. First, for each nest, which we implemented with data loggers, we removed all of the eggs from the nest, taking care to keep the eggs in the same orientation that they were found in within the nest. We took length, width, and weight measurements on each egg. We noted if they were banded, and if they had a band, we took the band length measurement at the widest point. Then when we were placing eggs back into the nest, we put a temperature data logger at the bottom of the clutch, the middle of the clutch, and the top of the clutch. We also put a soil moisture probe in the middle of the clutch, which collected soil moisture as volumetric water content. And then we dug a control nest near the real nest, placing a top, middle, and bottom temperature data logger at the same depths as they were placed in within the real nest. We then also placed an ambient temperature logger at each nest. So to analyze our data, we ran linear mix models to examine how temperature, soil moisture content, and metabolic temperature varied across the sand and marl nests. And then we determined metabolic temperature by subtracting the control nest temperatures from the real nest temperatures. So here you can see our mean daily temperature values over the course of incubation. So you'll notice here, there's a big dip in the temperatures, which occurred on June 3rd. That was due to a significant rainfall event that we had. This was the largest amount of rain that we received in this area over the incubation period. We received around 80 millimeters of rain, so that's why you see that big drop there. And the results from our analyses did not show that there was a statistically significant difference in the temperature values between the marl and the sand nests at any levels of the nest. So jumping to our soil moisture content, we had the inverse reaction, as you would expect when that rainfall happened, where we saw a spike in the soil moisture content within these nests. We did find that there was a statistically significant difference in the soil moisture content within the marl and the sand nest. You might be surprised, as I was, with that p-value. It's more moderately significant. I was expecting something more strongly significant. As you can see, there's a very strong visual difference here between the soil moisture content within these two nesting substrates, but basically what we believe it's not a stronger p-value because we had a low sample size. We only had 5 nests. But ultimately, we did conclude from this that soil moisture content was higher within the marl nest compared to the sand nests. So jumping to metabolic temperature, I first wanted to point out this blue line was one of our marl nests, and as you can see, it produced very low metabolic temperatures throughout incubation. This was the only nest of these 5 that failed, so that's why you're seeing that low metabolic heat generation. And so we did not include this nest in our analyses for the metabolic temperature. And so you can see here we did find a statistically significant difference in metabolic temperature when looking at soil moisture content. So we found that wetter nests had lower metabolic temperatures than drier nests. Well, the substrate type was not a direct predictor of metabolic temperature, it was an indirect predictor, as substrate type affected soil moisture content within nests, and the soil moisture content affected the metabolic temperature of nests. So jumping back to that nest that we had that failed, when we dug this up at the beginning of the season, we found that 80% of the eggs were banded. When we dug it back up at the end of the season to see what happened, we found that 5 of the eggs had embryonic development at varying stages, as you can see in that top photo. So this nest had viable eggs, so why did it fail? Jumping back to our soil moisture content figure, this blue line represents our failed nest. As you can see, after that spike in the rainfall, the rest of the nest, the soil moisture content decreased, whereas in our failed nest, it just kept increasing up and up and up and never stabilized. If you jump to our metabolic temperature figure, you can see that the metabolic temperature declined in all of the nests when the rainfall event happened, and then jump back up afterwards, except in our failed nest, where it declined and it just stayed down. So this tells us that this rainfall event really had a significant effect on this one nest and suggests that this nest failed due to soil moisture content within it. So we have observed that the marl substrate is not a well-draining soil, so we weren't super surprised that we saw higher soil moisture content rates within this nest. It does raise concerns, though, that during wetter years, we may see higher failure rates within these marl nests. In 2025, we found that 20% of our marl nests failed compared to only 5% of our sand nests in Cape Sable. And when we dug these up at the end of the season, we found that several of the marl nests showed signs of failure due to flooding. As the marl nests make up a large percentage of our total annual nests, for instance, in 2025, they made up 20% of our total annual nests. Throughout all of Florida. The crocs really love nesting here because it provides very good nursery habitat, the canals. But this just really shows you why it's important to understand what's going on within these nests. So we will be continuing this study in 2026. We have already implemented 3 sand nests in East Cape Creek with data loggers, and we will be instrumenting 3 marl nests with data loggers next week when we get back to the U.S. I'd like to thank the U.S. Fish and Wildlife Service and the National Park Service for providing us with the permits to conduct this work, and our entire CrocDocs team for assisting with fieldwork and data collection. Thank you.
Thank you, Brenna. And right on schedule. Okay, hang on a second. Another one that's eager before I call her. Give me a second. Okay, our next speaker is Melina Simoncini from Argentina, and today she's going to tell us about Cayman latirostris.
Good afternoon. I am Melina Simoncini. I'm a researcher from Argentina. And today I will present a study entitled Between 31 and 33 Celsius degree, how temperature modulates sex ratio in Cayman latirostris. In Cayman latirostris, sex determination is temperature dependent, and both constant incubation temperature and temperature fluctuations influence the sex ratios and the hatching characteristics. Given that in constant temperature incubation in 31 Celsius degree produce 100% females, and at constant temperature at 33 Celsius degree produce 0% or 0% of females or 100% males. Our hypothesis was that alternating incubation between 12 hours at 31 Celsius degree and 31 at 33 Celsius degree would produce approximately 50% females. In addition, we tested other combinations of exposure to these constant temperatures. Okay, in order to evaluate how temperature oscillation affected sex ratio and embryo survivors, we incubated a total of 15 clutches of Cayman latirostris in lab over 4 reproductive seasons. We separate the clutches in different temperature treatment, but to the exposure time at the 31 and the 33 Celsius degree. If you can observe in the figure, we here have a 33 Celsius degree for 20 hours, here 31 for 24 hours, and different time to expose the temperatures. Okay, our results shows that as the hours of incubation at 31 Celsius degree increase, the percentage of females increase too, following the Sigma curve. It's possible to see in the figure that the curve. And the sex ratio was different among different clutches, and the deviation decreased as increased the number of the hours for each temperature. In the constant temperature, we didn't have variation, and in the middle treatment, the variation is more. Although we expect to obtain 50% females at the 12 hours at 31 and 33 Celsius degree, the average female ratio approximately 72% of female. That recorded in previous study when incubated at constant temperature of 32 Celsius degree. The embryo survivor was similar between incubation treatments. In conclusion, based on our results, we can say the pattern of export to different thermal regimen has no linear effects on sex. In future study, we will incubate clutches at constant temperature of 32 Celsius degree and alternate it between 12 hours at 31 and 12 hours at 32 Celsius degree in order to evaluate whether the temperature oscillation produce sex ratio equivalent to those obtained at a constant average temperature. Thank you very much for your attention.
Melina, you have like 5 more minutes. Thank you, Melina. Another eager one before I call him. Here we have, Carlos is going to tell us about overposition climatic conditions in Cayman latirostris.
Good afternoon to everybody. Happy to be here with you, and I want to congratulate the organizer of this wonderful meeting. It was such a pleasure to be for the first time in my life in Morocco. I will talk this presentation, which is part of Florencia Valley PhD dissertation. Unfortunately, she couldn't make it to Agadir to be here with us. The title is Pre-oviposition climatic conditions are related to offspring sex ratio through maternal effects in Cayman latirostris. So far, we know that Cayman latirostris present temperature sex determination. Menelia already mentioned that. We have a thermosensitive period that occurs between stage 19 and 24. We know that 31 produce 100% females, 32% and 100% males, and incubation at 32 produce females and males, 70% females, 30% males, but it depends a lot of the nest. Some nests will produce 100% females, half of them will produce like 50-50, and just a few of them will produce more male than females. So we were thinking that what was going on those nests, what was happening to have such a difference. And on Florencia PhD, we work trying to identify if there was some kind of climatic variation or if there is some kind of information from that year that could influence on the sex ratio of the clutch. Our objective was to evaluate the effect of intra-annual climatic variation on maternal investment, on steroid hormones, and fatty acid, and the relationship with the sex determination. We collect eggs in different parts of Santa Fe province. We collect 21 nests over a period of 4 years. We measure climatic variation at the end of the reproduction season, also at the very beginning, and that is because at that time the females start to uptake resource for the next reproduction, and then at September, October, in our spring, they start to produce vitellogenin and produce the eggs. Then, when we collect the eggs, we have a sample of the yolk in order to determine the fatty acid and estrogen and progesterone. And when they hatch, we took the sex of the animals plus the morphometrics. Every egg was counted to be they were fertile, they were measured, and all the eggs we used in this experiment were younger than stage 5 of incubation. The eggs were placed on vermiculite and plastic container and set up to an incubator under 32 degrees of temperature, and it was constant temperature. The temperature was recorded with a hollow device, and when they hatch, we measure the animals. The climatic variable were obtained with Google Engine, and we measure fatty acids, steroid hormone, and as I mentioned, we sex the animal. The animals, we sex 5 of each clutch because unfortunately we have to do gonadal inspection to be sure, and we have to kill the animals, so we want to reduce the number as much as possible. Over the 4 years of the study, we found that 2020 and 2021 were dry, but not as much as what happened in 2022 and 2023. And that happened in the north of the state and also in the center of the state, which cover all the distribution where we collect the eggs. We found relationship between the mean temperature in March and the amount of testosterone. When I mean testosterone, I mean inside the eggs. We found a relationship also with testosterone and the accumulative precipitation in September and October, and it was negative from testosterone with, I'm sorry, reading, fatty acids. Oh, shit. Palmitic and palmitoleic acid were related to the percentage of female, inversely related. The number of female increased at the temperature in September and October increased. And mean temperature in September and October were inversely related to the fatty acids. I mentioned mean temperature in March was related to testosterone. The cumulative precipitation in September and October also was related positive to testosterone, but mean temperature in November was inversely related to testosterone. On the other hand, we did not find any kind of relationship on estrogen. We know that testosterone have variation and it responds to stress condition. What we found that the highest level of drug, as it gets more severe, it produces a reduction on testosterone. That could be a relationship that some female, maybe the best one, are reproductive active on those years, and that could be some kind of connection to the percentage of the sex of the individuals. In 2020 and 2021, remember those were dry year, but not as dry as what happened in 2022 and 2023. The percentage of female dropped by half. You know, as we get worse situation, low rain, the percentage of female was reduced. But remember, that is something that happened previous to the incubation, that the female is producing to the clutch, that then under a 32 incubation temperature, constant, did change a lot from 76% in 2020 and 2021 to 32 and 31 on the next couple of years. We think that the proportion of female have been reduced during La Niña event, which is dry year in the wild. When we get La Niña event, Melina's work has shown that the proportion of female is also reduced, but that is in the wild under natural incubation. But even though when we control the temperature, it also is reduced. So what do we have? Increase in palmitic acid and palmitoleic acid are associated with a reduction in females. Climatic condition prior to the oviposition are associated with the percentage of female, fatty acid, and testosterone, but not estrogen. And as we get more drug in consecutive years, the proportion of females is reduced. These are, I mean, it's not cause effect because we are not controlling the variable, but we are planning to do so in the near future to try to change the diet of the female, increasing some fatty acid, and see if that in fact indeed influence sex determination. We're going to assess the relationship between female body and physiological condition, and how do they transfer estrogen and testosterone to the yolk, and finally identify other maternal components that could influence sex determination at temperature that produce both sex. Thank you very much, and I think we will have time for questions later on the session.
Thank you, Carlos, for your student. Yeah, you can, no, we go. We're not going to do the panel discussion right now. We still have 3 more speakers, but we have a break. You guys can, no, después. We have a little break for about 20 minutes. While as you are leaving and coming, you're going to see a couple videos up here. It's Crocodile. One was done in the Northern Territory of Australia. You'll see it's really brilliant if you haven't seen them before. And then there's one from Malagasy, and Christine can always come tell us about it as you guys are coming in and out. But go get something to drink and come on back. Be back in the next 20 minutes. We will start promptly at 3:15.
Session. Okay, so you want to put that in after I do this now.
You gotta be clockwise if you're hanging near the water. Keep a sharp eye on your son and daughter. If you fish from a boat, keep away from the side, cause the crocodile's mouth gonna open up wide. Don't hunt by yourself if you're fishing with a spear. Always make sure that someone else is near, cause you don't have eyes in the back of your head, and if a crocodile's behind you, you might find yourself dead. In the rivers of the sea, there are crocs unseen. Now there's big and small, and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. Near the river or a crossing where the crocodiles are, you might see a sign if you're driving in your car. Take heed of the sign if it says not to swim. Don't risk your life on a dare or a whim. Crocodiles can move around to any waterway. What was safe to swim the other week might not be safe today. Even if you think that you know it's safe to swim, you're gonna risk your life if you jump right in. In the rivers of the sea, there are crocs unseen. Small and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. Make sure you tell the little ones to be croc-wise. Let them know of the dangers and how to read the signs. Let them know that in the past some people have gone missing, maybe taken by a crocodile or swimming or fishing. In the rivers or the sea, there are crocs unseen. Up and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head. In the rivers or the sea, there are crocs unseen. Not as big as mouth and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see.
Be crocwise means knowing that saltwater crocodiles are dangerous and can live in any river, billabong and ocean. A long time ago, there were lots of crocodiles. Then people started to hunt and kill crocodiles for their skins. Because of that, in about 1970, there were only a few crocodiles, not like before. In 1971, people stopped killing crocodiles and there became more and more of them. Now there are lots of crocodiles, like a long time ago. Now lots of crocodiles are swimming around everywhere and traveling very far to find new places to live. Those crocodiles might be living in water where people did not see them before. So remember,
In the rivers of the sea, there are crocs unseen. Now there's big and small, and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see.
It's called closing ceremony. So basically, it goes straight after your after your session, with your session, whatever, because we don't have another break or anything. Thank God.
I presume this is the one that Matt said to remind him to play a video.
To what, sorry?
Matt told me to remind him to play a video. That must be it.
No.
Another video?
He said a Facebook video that. Okay, I thought that had gone so that we actually. We have to chase. No, I think so too. Like, it's a Facebook link, so.
Don't worry about it. He should be in here. And if he's here, I'll ask him. But
like we'll just move on. We'll probably.
How long is the video? Closing ceremony?
Oh, it's not a video, it's a PowerPoint.
Okay.
And it's just got points at just thanking
okay,
people
cool.
and yeah. So Charlie's a bit annoyed I've put it together, but I don't know. We've got friction in the ranks. Use it, don't use it, I don't care. It's there if you want it.
We can turn that one off. Although I like having the music going in the background. No, the music is that one.
I struggle.
You gotta be clockwise if you're hanging near the water. Keep a sharp eye on your son and daughter. If you fish from a boat, keep away from the side, cause the crocodile's mouth gonna open up wide. Don't hunt by yourself if you're fishing with a spear. Always make sure that someone else is near, cause you don't have eyes in the back of your head, and if a crocodile's behind you, you might find yourself dead. In the rivers of the sea, there are crocs unseen. Now as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. Near the river or a crossing where the crocodiles are, you might see a sign if you're driving in your car. Take heed of the sign if it says not to swim. Don't risk your life on a dare or a whim. Crocodiles can move around to any waterway. What was safe to swim the other week might not be safe today. Even if you think that you know it's safe to swim, you're gonna risk your life if you jump right in. In the rivers of the sea, there are crocs unseen. Not as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. Make sure you tell the little ones to be croc-wise. Let them know of the dangers and how to read the signs. Let them know that in the past some people have gone missing. Maybe taken by a crocodile or swimming or fishing. In the rivers of the sea, there are crocs unseen. Not as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. In the rivers of the sea, there are crocs unseen. Not as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see.
Be crocwise means knowing that saltwater crocodiles are dangerous and can live in any river, billabong and ocean. A long time ago, there were lots of crocodiles. Then, people started to hunt and kill crocodiles for their skins. Because of that, in about 1970, there were only a few crocodiles, not like before. In 1971, people stopped killing crocodiles, and there became more and more of them. Now there are lots of crocodiles, like a long time ago. Now lots of crocodiles are swimming around everywhere and traveling very far to find new places to live. Those crocodiles might be living in water where people did not see them before. So remember,
rivers of the sea, there are crocs unseen. Now as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see.
be crocwise.
You gotta be crocwise if you're hanging near the water. Keep a sharp eye on your son and daughter. If you fish from a boat, keep away from the side, cause the crocodile's mouth gonna open up wide. Don't hunt by yourself if you're fishing with a spear. Always make sure that someone else is near, cause you don't have eyes in the back of your head, and if a crocodile's behind you, you might find yourself dead. In the rivers of the sea, there are crocs unseen. Now as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. Near the river or a crossing where the crocodiles are, you might see a sign if you're driving in your car. Take heed of the sign if it says not to swim. Don't risk your life on a dare or a whim. Crocodiles can move around to any waterway. What was safe to swim the other week might not be safe today. Even if you think that you know it's safe to swim, you're gonna risk your life if you jump right in. In the rivers of the sea, there are crocs unseen. Now as big as small, then they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head, see. sure you tell the little ones to be croc-wise. Let them know of the dangers and how to read the signs. Let them know that in the past some people have gone missing, maybe taken by a crocodile or swimming or fishing in the rivers of the sea. Then the crocs unseen. Now the biggest up and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head. In the rivers of the sea, then the crocs unseen. Now the biggest up and they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head. See.
Be croc-wise means knowing that saltwater crocodiles are dangerous and can live in any river, billabong and ocean. A long time ago, there were lots of crocodiles. Then, people started to hunt and kill crocodiles for their skins. Because of that, in about 1970, there were only a few crocodiles, not like before. In 1971, people stopped killing crocodiles, and there became more and more of them. Now there are lots of crocodiles, like a long time ago. Now lots of crocodiles are swimming around everywhere and traveling very far to find new places to live. Those crocodiles might be living in water where people did not see them before. So remember.
The rivers of the sea, there are crocs unseen. Now there's big and small, but they're moving upstream. Crocodiles are deadly, just remember that they're deadly. If you're hanging around the water, you gotta use your head. See.
Be croc-wise.
Ethan. Nice to meet you. Should I sit up here?
Yeah.
Okay.
And then when I'll call you up.
All right.
No need to rush the stage.
Yeah, that sounds
And then I'm
good.
going to be sitting there with Tom.
All
Okay.
right. I'll try to resist.
I know. Everybody's so excited about it.
All right, are we ready for the remaining afternoon session? I'll ask everybody to take their seats. Okay, our first speaker for the Crocodile Biology Session Part 2 is going to be Daniela Neres Nosa. Daniela, she's going to tell us about broad-snouted caiman populations in Brazil.
Hello everyone again. For the ones who wasn't before, my name is Daniela and I'm the coordinator of the sanitary and ecological monitoring of the Caiman program, initiative of the Marcos Daniel Institute. Is located in southeastern Brazil. These studies involved collaboration among researchers from different institutions, including universities, conservation organizations, and research centers. To begin, I would like to briefly introduce the environment context of this study. In 2015, the Fundão mining dam collapsed in southeastern Brazil. This event caused major environment changes across the Rio Doce basin, affecting aquatic and riparian habitats along a large spatial extent. However, the dam collapse was not the only pressure affecting this region. The Rio Doce Basin has also been historically influenced by the other human activities, especially agriculture, urban expansion, habitat fragmentation, and pollution. 10 years after the disaster, it remains important to understand how aquatic and semi-aquatic wildlife persist in this impacted landscape. I don't know what's happening. Okay. So in this context, protected area may play an important role. Protected remains of the Atlantic forest can function as a refuge for biodiversity, especially in landscapes affected by multiple environment pressures. These forest areas may provide more stable ecological conditions, including better habitat structure, lower disturbance levels, and more suitable environment for aquatic and semi-aquatic species. For the crocodilians, protected wetlands, rivers, lakes, and flooded areas may be essential for population persistence. In this study, we especially focus on broad-snout caiman, the Caiman latirostris, and evaluated, however, protected habitats may contribute to maintain its occurrence in the real dose basin. This slide shows the spatial design of our feed surveys. The Rio Doce Basin extends across the southeastern Brazil from upstairs region. Okay, so from upstairs regions in Minas Gerais to downstream areas and the Delta region in Espírito Santo state. The map shows that basin outline the survey sites and the location of the legally protected Rio Doce State Park. So this is Rio Doce State Park. Between 2024 and 2025, we conducted 109 nocturnal spotlight surveys. These surveys cover different portions of the Rio Doce Basin, from upstream areas to downstream river sections. This allowed us to assess caiman presence across different areas with different levels of protection and human pressure. This approach was important because the persistence of aquatic species in impacted basin is not determined only by the original disaster, but also by the current conditions of the landscape and the availability of suitable habitats. We used nocturnal spotlight surveys to detect caimans through eye shining. When caimans were observed, we recorded their occurrence, and when was possible, classified them as juveniles, subadults, or adults. After the field surveys, we compared caimans' occurrence between protected and not protected areas using a generalized linear mixed model. We also calculated encounter rates as individuals per kilometer, always considering possible variation in the detectability. For example, the vegetation cover, water levels, weather conditions, observer visibility, and habitat structure may influence the probability of detecting caimans during surveys. Therefore, encounter rates were used as a comparative ecological indicator, but not as an absolute estimate of the population size. Broad-nosed caimans were recorded across different portions of the Rio Doce Basin. On the map, the yellow circles indicate seats where caimans were detected during this study, and the red circles indicate seats where caiman were not detected. The white sarge icons, like those, indicate areas where solid waste or salt were observed during the surveys. These records indicate that the species is still present in different portions of the basin, including areas outside of the Rio Doce State Park. However, the pattern of the detection was not homogeneous across the landscape. The records included juveniles, subadults, and adults, suggesting that the species still maintain population structure in some areas. One of the clearest findings of this study was the contrast between protected and not protected areas. In non-protected seats, detection were rare, occurring only 90.5% of the surveys. These results suggest that protected areas play a central role in maintaining the occurrence of the broad snow caiman. The contrast also indicate that the habitat protection may be especially important in landscapes where aquatic environments are affected by the multiple pressures. Encounter rates were higher inside the protected area, with 1.5% individuals per kilometer. In non-protected sites, the rate was much lower at 0.02 individuals per kilometer. This difference suggests that the protected habitats may support higher caiman's occurrence and possible more suitable conditions for the population persistence. However, as I mentioned before, these values must be interpreted with caution because the technicity may vary among sites and environment conditional. Even so, the difference between protected and no protected areas was consistent with the occurrence data and supports the importance of protected forest habitats for aquatic biodiversity. Our results indicate that protected Atlantic forest habitats are important for sustained broad snow caiman population. These areas may function as a refuge for aquatic biodiversity in a heavily impacted base. I'd like to thank the Caiman Program, the Marcos Daniel Institute, and the Brazilian Biodiversity Fund for supporting this research. I also thank you all the partner institutions, researchers, field team members, and collaborators who contributed to this field surveys, data analysis, and conservation action. So I finish saying that caring for nature is caring for the continuity of our own lives, and we cannot forget that. Thank you very much.
Thank you, Daniela. There's going to be an addition to the program. We have a last-minute secret talk that's going to be coming in after the third talks. We're actually going to have 3 more instead of 2 more. So sit tight. You guys are in a good spot. I now have Ethan Sheely coming up. Ethan is a grad student at University of Georgia. And if I can get his slideshow up. All right, here's Ethan.
Hey there, everybody. Yeah, for those of you that don't know me, my name's Ethan Sheely. I'm a PhD student out at the Savannah River Ecology Lab, working with Ben Parrott and his research group. And today I'm going to be talking about some work that I've done as part of my dissertation there on trying to develop an age estimation tool in the American alligator. So I'm sure anyone that's worked with, large crocodilians knows that aging them morphometrically can be really hard, if not impossible. Just looking at an animal like this is Stumpy. He's a captive gator that lives at SREL. He was attacked by a nuisance human back in Alabama in the 1980s, and so he was relocated into captivity. And in 1982, when they brought him to SREL, he was about 2 3/4 of a meter long. The next time he was measured was in 2006, and he was at 3 3/4 of a meter long. And he hasn't grown an inch since then. This is just some evidence that size is not really a good marker for age in these animals. I'll talk about it a little later, but it seems like at least in alligators in these South Carolina populations have determinate growth. And so why do we even care about age independent of size? Well, it enables us to do a lot of things that can be really useful for both conservation and management. But using age of individuals within your population, you can look at the age structure along with temporal trends. So how is recruitment every year in your population? as well as developing survival and age-dependent mortality estimates. So at what age do most individuals in your population die? How long are they reproductive on the landscape? And identifying age at reproduction and whether there's any age dependence to their fecundity. And so all of this can really inform conservation and management decisions. So if you're monitoring the recovery of depleted populations, then again, knowing things like recruitment is really useful. And it can also advise sustainable use practices based on population age strata. So knowing which, how many individuals you have of a certain age that you may want to harvest or manage. And finally, it can also just increase our biological understanding of these animals. I think there's a lot that's unknown about the life history of crocodilians, such as whether they actually ever die of just old age, known as negligible senescence. And also little is known about how age interacts with other things such as contaminant burdens or disease susceptibility and age-related reproductive success. So I really feel like there's a lot that we can learn from this. And we'd also know that, you know, just generally crocodilians can live a long time. This is some data I visualized from this Weigel 2014 paper of some maximum recorded lifespans of captive crocodilians. And you can see that they easily reach up into the 60s and 70s, and pretty much all of those individuals were both received as adults, so you could probably tack on another 10 to 20 years onto those, and most of them were still alive at the time this data was published in 2014. So some really long-lived animals across the whole taxonomic group. And so there are some current methods of age estimation. So like I said before, people in the past have used things like size-based age estimation. There can be a lot of error in this due not just to this process of determinate growth, but also environmental factors and population-specific differences can really change the way that these animals grow. And then also another method that's been explored is this thing called skeletochronology, which is using cross-sections of bones, basically similar to how you age a tree based on its rings, based off of alternating growth and arrested growth periods based on seasonality. But this has a lot of drawbacks to it. It requires really invasive sampling. You have to remove a bone from the animal. And there's limited validation and really noisy data that's usually produced by this. And plus, in a lot of crocodilians' ranges, there really aren't warm and cold seasons. So these patterns of growth typically aren't universal. And so what we can look to is actually human health science. So there's actually a ton of research that goes on aging of humans, of course. We want to figure out how to, nip that stuff in the bud. So, there's all this technology, especially in the genomics and molecular world, that has been found to be biomarkers of the aging process among humans. And one that's the most studied at this point is something called DNA methylation, and these models that are produced using this called epigenetic clocks. And I'm not going to get into a ton of details here or go over my methods that much in depth, because you can come find me later and I'll talk your ear off about them if you want. But in its simplest form, DNA methylation is a chemical tag that's attached to the DNA strand at certain locations in the genome, and it's used to do things like regulate which genes are turned on and off. But it's been found that certain places in the genome, these DNA methylation marks are lost or gained at very constant rates across individuals. And so you can combine a number of these markers to create predictive models of age. So this is from a really large human cohort. You can see the actual age of those individuals along the x-axis and their estimated age based off of their DNA methylation on the y. And it can be really surprisingly accurate. And there's actually quite a few species at this point that this has been done in aside from humans. Mostly mammals. There's actually a universal mammal aging clock because there's enough conservation and we have enough sequence data from across all these mammals to be able to do this. And so, but it's also been done in a handful of fish, a lizard, some birds, and xenopus frogs. So there's really no reason that this technology can't be extended to other species such as crocodilians. Yeah, and a similar pan-crocodilian approach could even be possible if this kind of research was given enough resources. But to build these models, the hardest thing that you have to do is collect a data set of known age individuals to calibrate your model with. You need to have as much of the lifespan age range as you can and as many individuals as you can, because that will only improve the accuracy. So this is just a little map of where we got most of our samples from so far. These are a mix of both captive and mark recaptured individuals from the wild, trying to make it as robust to both of those things as possible. And here's the distribution of ages that our data set contains, as at the time that we did a bunch of sequencing on these. So we have about 35 individuals ranging in age from hatchlings to 36 years. And I'd like to give a huge shout out to the people that actually contributed samples to this. This has been a really collaborative project, really having to reach out and communicate with a bunch of different people. So huge thanks to everyone up here and all these institutions, and especially the St. Augustine Alligator Farm. They provided a ton of our samples and have great records on their animals, and were really willing to help. So yeah, huge shout out to them. But just looking at the actual model here a little bit, if you want to understand what actually goes into this, it's fairly simple. So each one of these little plots here is one of those locations in the genome where the frequency of DNA methylation is changing. And you can see that each one tends to either decrease or increase across these different individuals, and it's at a pretty constant rate. And if you combine enough of these, in our case 56 of these sites, you can create this predictive model. So this is ours. Again, the actual age of the animals is on the X and the predicted age is on the Y. You want to pay attention mostly to the red dots. Those are individuals that our model has never seen before. It was trained on the black dots, and then red dots were held out for this validation. And the accuracy is quite good, out to 36 years, which is the oldest individuals in our data set. And you can see the average error there is around plus or minus 3 years in this model. And so for our next steps, what we want to do is develop a more targeted assay. So right now, the method we're using requires whole genome sequencing, which is really expensive. And you're really generating a lot of unnecessary information from that if you're just interested in these specific sites. So these targeted assays based off of PCR can measure only the sites that you're interested in, and you can get the sample cost per sample down quite low if you optimize it enough. And so to really broaden this as much as we can and make it more practical, this is going to be our goal. And I've made some good progress on this. I've picked out 10 sites across the genome that I've successfully amplified with PCR. You can see some of the bands in the top right there, and these are all able to go into a single reaction, like a multiplexed PCR reaction. And you can see that we lose a little bit of accuracy using this method versus whole genome. The error rate goes up to about 4 years on average instead of 3, but still very reasonable, I think. And so our other next step that we want to do is expand our testing set. So not only do we want to replicate our results from the whole genome sequencing with this new PCR approach, but we also want to sequence some additional animals that we've acquired since our initial sequencing, especially some old individuals we have. We have quite a few individuals now that we didn't have an exact hatch date from them. Most of them are from mark recapture studies that were marked way back in the day as adults. So we have at least minimum known ages on them. So for example, Bertha here is from Yawkey Wildlife Center in South Carolina, was marked as an adult female at over 9 feet. She was a huge female too, in 1981, and she's still nesting there today. So we have a couple sequential samples from her over the years. And then this other animal is Cowboy. He's even older, but was an adult male captured by Tom Murphy in 1973. And so if you take anything away from this, I just ask you to put your thinking caps on for a second here. Even if I were to trip off the stage and break my neck right now, this technology will be available in crocodilians within the next 5 to 10 years. I would be really deliberate about thinking about how being able to age an animal will change the way you're conducting some of your research, some of your management practices, your conservation efforts, and even things like captive animal husbandry for those zoological institutions. Additionally, we're always looking for additional samples of known age crocodilians, whether they're alligators or other species. Right up there is my email address, but really all we need is a sample of blood. It can be red blood cells. We're not really picky about how it's been stored as long as it's still in fairly good shape. It's really easy to get DNA from. And so there's my email address, and that QR code will take you to our lab's website that also has some of our contact information. And with that, I'd like to acknowledge my advisor, Ben, and the rest of my lab, as well as this informal alligator aging consortium of all these people that have contributed samples at this point, and my funding sources, including the CSG itself. I recently received the SRAS grant for contribution to this work, so that's been really key in keeping this project going.
Thank you very much, Ethan. That was great. No falling off the stage, anybody? Okay, another, okay, I see him, he's coming. So originally this was going to be the last speaker for today, and I was going to introduce him as, well, you know, you always got to go out with a bang, but here is a legend in and of himself, and we will hear all about Joe's travels around the world and the experience that he has with American crocodiles. So without further ado, here is Joe, and please stay through for the next speaker.
Thank you, Benicia. Well, here we are. We were supposed to be the last show. They said we're going to finish with the bang. Anyway, I just want to talk. I've actually been working with American crocodiles for about 40 years, and with many people here in this room and some that are not. And my mantra is to train all these young guys and girls and then let them get their education and everything, and then I want to work for them. That's what I want to do. So without going any further on that, let me just talk about, you guys can read that while I talk. I have two institutions that I really have to thank for my career. One of them, I worked for Florida Power and Light for 30 years. The other one is allowing me to take all of this knowledge and take it around the world, and that's Gatorland Global. So both of those have been instrumental, plus a ton more. But I just want to, we've been talking about American crocs for the last 4 days, so I'm just going to go through these slides. I'm only going to talk about 2 countries that I've worked in with Acutus, and we know that's the range. We all know that. We don't have to go over that. This is something I talked about in El Salvador, is we've all caught crocodiles here. Have you guys ever noticed the scutes, the scales? I'm sure we all do. And do you know that this is the typical pattern for Acutus, and this is the typical pattern for Niloticus, okay? However, there is a kind of a shot of a live one with the typical pattern. However, their Acutus is everything but natural.